These tiny, beautiful wasps eat the hearts out of cockroaches. Jewel wasps carve up cockroaches like jack-o'-lanterns in a way scientists have never seen before. If you loathe cockroaches, you are going to love the emerald jewel wasp. Females of the species Ampulex compressa, known also as emerald cockroach wasps, are less than an inch long, and decked out in gorgeous, metallic green blues. To complete their life cycles, they must first sting an American cockroach and inject the much larger insect with mind control toxins that turn it into a defenseless zombie. Next, the female wasp drags the subdued cockroach by its sensitive antennae into a cavity it has found, lays a single egg on the roach's leg and then uses dirt and debris to seal the fateful pair inside. After 6 days, the egg hatches and the larva carves its way inside the cockroach's chest. It then begins devouring the helpless insect from the inside out. It is one of the more macabre horror stories you will find in nature, and just in time for Halloween. Gruesome as it is, there are around 500,000 species of parasitic wasps on this planet, and many make their living in a similar fashion. Scientists love to study these mini monsters because many wasps zero in on insects that humans don't really mind seeing brutalized. Creatures like roaches, but also crop pests and invasive species. But there's something scientists never noticed about the jewel wasp's cask of amontillado routine until now. Since the 1800s, people have sort of had this mantra that parasitoids selectively avoid eating the vital organs of their host so that they can keep it alive, said Kenneth Catania, a neuroecologist at Vanderbilt University. And what I have found is that this parasitoid goes straight to the heart of the cockroach and eats it. Each fall, Dr. Catania prepares a special, Halloween-themed class for students of his Neurobiology of Behavior course. One year, he even built a 3-room, Wes Anderson-style diorama and filmed a wasp dragging its victim through a dollhouse-size kitchen full of tasty, tiny treats and into a tomb shaped like a human skull. To be clear, the video is meant to be silly and engaging, the better for students to learn how the wasps operate. But behind the cutesy props, it also shows real ecology in motion. The wasp performs her task perfectly, even going so far as to seal the skull's eye socket with tiny bits of golden plastic treasure, just as Dr. Catania hoped she would. And so, in another attempt to win his students' attention, the scientist set out to film an emerald jewel wasp larva as it feasted on the cockroach from within. That's the way science often unfolds for me, said Dr. Catania, the author of great adaptations. I'm looking at something out of curiosity or art. This is how he ended up capturing the larva's taste for cockroach heart. But he made an unexpected discovery. After eating the heart of the cockroach, the wasp larva started gnawing at its quarry's trachea, the insect equivalent of lungs. This caused air to leak out of the cockroach's respiratory system and into its body cavity, air that the wasp larva then eagerly slurped up. In other words, the emerald jewel wasp both eats the cockroach's heart out and takes its breath away. After performing the experiment 24 times, Dr. Catania was able to show that not only do the air bubbles allow the larva to breathe while fully inside the cockroach's body, but they also give the little hellraiser a metabolic boost. Once the air bubbles appear, the larvae start to chew faster, which Dr. Catania documented this year in a study published in the journal Current Biology. Within 48 hours, the emerald jewel wasp larva has chewed its way through so much cockroach that the host dies. You can imagine it sort of like Alien, said Dr. Catania, referring to Ridley Scott's 1979 film. Once it's inside you, it's game over. In fact, the game ends much faster than has been observed with other parasitoids, some of which don't even fully kill their hosts at this stage, but rather keep their victims on life support by avoiding vital organs. That led Dr. Catania to ask why these wasps evolved to consume with the speed of Joey Chestnut. That sets up a perfect cliffhanger for a sequel to this natural slasher flick. Dr. Catania believes the wasp has to eat quickly before its zombified host is eaten by, well, something else. But that's a discovery he's not yet ready to publish. "There's more to this story," he said, "and I'm currently working on the second part." In the 1880s, Henry Faber was captivated by the special art of eating, whereby a parasitoid wasp larva fed selectively on host internal organs, avoiding the heart, dorsal vessel, and tracheal system, respiratory system, to preserve life. In Faber's words, the ruling feature in this scientific method of eating, which proceeds from parts less to the parts more necessary to preserve a remnant of life, is none the less obvious one. Subsequent investigators have reported the same for many parasitoid wasps too, including for the emerald jewel wasp, Ampulex compressiva. Here it is reported that larval jewel wasps destroy the dorsal vessel and tracheae, respiratory system, in the thorax of their cockroach host, Periplaneta americana, at their earliest opportunity. Moreover, the broken tracheae release air into the host, which the larval jewel wasp inspires. An increase in larval chewing rate, cotemporaneous with the sudden release of air from the host's broken tracheae, suggests the larva taps into the host respiratory system to support its metabolism while rapidly consuming the host. To put the relative timing of these events into context, it helps to first consider the stages of wasp larval development. 5-6. The wasp egg hatches 3 days after oviposition on the cockroach middle leg, and the larva feeds on hemolymph through a hole chewed through the proximal coxa, upper leg. On day 6 after hatching, the larva begins entry into the cockroach mesothorax, middle thorax, by enlarging the hole and squeezing through. When the larva reaches the midline of the mesothorax, it consumes the mesothoracic dorsal vessel and corresponding thoracic chamber of the heart, figure 1c. Video S1, clip 1. Shortly thereafter, it enters the metathorax and consumes the metathoracic section of the dorsal vessel and metathoracic chamber of the heart, figure S1 in the supplemental information. The mean time between first appearance of the head and mandibles at the dorsal margin of the mesothorax and the destruction of the dorsal vessel and heart chamber in the mesothorax was 156 minutes for 10 cases. Although the caudal, abdominal, heart chambers continue to pump after these more rostral chambers are destroyed, the heart can no longer pump hemolymph to the head or first thoracic segment. Given that the larva does not pupate for roughly another 2 weeks. 7. The destruction of the heart segments in the thorax occurs early in the sequence of larval development. In each of 24 cases observed, the larva also chewed through large respiratory tracheae as it entered, often resulting in the release of air, figure 1d. Video S1, clip 1. The larva always entered the cockroach such that its rostral spiracles on one side were oriented dorsally, with respect to the host, where pockets of air were often located. As a result, there was often prolonged contact between larval spiracles and air pockets. These air pockets, in turn, were often still attached to the host's severed tracheae, expanding and contracting in apparent coordination with host respiration, figure 1d. Video S1, clips 2 and 3. This provided a link between the respiratory system of the host and the parasitoid. Moreover, air from the host's broken tracheae created isolated bubbles. When one of the larva's first or second spiracles encountered a bubble, the bubble was usually inspired. This remarkable and consistent behavior, figure 1e. Video S1, clips 4 and 5, clearly shows the larva's spiracles are active within the host. At the earliest stages of host consumption described here, the larva's caudal spiracles have yet to be drawn inside the host, but as the larva moves progressively further into the host, the remaining spiracles are necessarily drawn progressively into the host's body. Hence the distance from the active mandibles to the dwindling free spiracles increases with each increment of larva ingress until finally the larva's entire body is inside the host. Video S1. To investigate the possibility that air derived from severed host tracheae serves the larva's metabolic needs, cases were identified in which air did not leak into the mesothorax to create an air pocket despite severed tracheae. In these cases, subsequent movement of the larva into the metathorax while severing additional tracheae eventually caused leakage of air into the host body cavity. In 4 such cases, it was possible to sample the cyclical chewing rate for repeated, 5-second intervals when the larva mandibles were periodically visible, both before and after an air pocket had suddenly formed around the head and rostral segments of the larva. For each case, there was a statistically significant increase in chewing rate after the air pocket formed. Figure 1G shows a case in which the larva mandibles were visible almost continuously before and after the formation of the air pocket, allowing for a clear graphical representation of chewing rate versus time, relative to air pocket formation. See figure S1 for more cases. Note that as the larva continues to feed, moving into the abdomen while breaking additional tracheae, the air pocket usually expands to eventually surround the entire larva at later stages. The results show that the larva destroys portions of the dorsal vessel and tracheae of its host at a comparatively early stage relative to many other parasitoids. 8-9. Weather.